Everyone. We are going to talk about reproductive technology, genetic testing, and gene therapy this week, Chapter 16. So in 1978, a woman named Louise Brown was the first human born after in vitro fertilization, the procedure which an egg is fertilized outside of a woman's body. In vitro literally means in glass, and then the embryo is implanted into the woman's uterus to complete its growth. Successful reproduction requires many, many things. We've learned that the reproductive system is very complicated. So successful IVF rounds require healthy gametes, both the egg and the sperm, a place for fertilization to occur, like a woman's oviduct, a place for the embryo to grow and develop, the woman's uterus. There are a couple different types of infertility. Primary infertility is trouble conceiving a first child, and then anything after that is considered secondary infertility. Causes: 40% is the female's issues, 40% is male issues. So it's about even there. 20% is unexplained. And then other things like substance usage, alcohol, tobacco, age, and weight also play a factor in infertility. When females are infertile, there are a few different reasons why. Hormones are to blame a lot of the time. Proper estrogen levels are required for ovulation. Some women have ovarian damage, oviduct blockage, or uterine or cervical problems. So women have a lot of different parts that need to be working properly in order to be fertile. Infertility in women increases with age. We lose our ability to host children, to host embryos, to healthily host embryos as we get older. Male factor infertility. Sperm defects. There can be a lot of things wrong with a male sperm. Low sperm count, low sperm motility, absence of sperm. Some men don't produce sperm at all. That's called azoospermia. Or a Y chromosome defect may be associated with male infertility. There's the chart of male factor infertility, what could be wrong. And a quarter of it is just unknown. Assisted reproductive technologies is the whole umbrella. There are various ARTs that have expanded childbearing options in the last 30, 40, 50 years. So intrauterine insemination with a donor sperm is one. Egg retrieval combined with IVF, egg donor combined with IVF, in vitro fertilization and related procedures, and then surrogacy. We will talk about all of them. So artificial insemination and embryo transfer. This happens if the father is infertile, the mother is inseminated by a donor and then carries the child. If the mother is infertile but able to carry the child, they can get a donor egg and then inseminated by the father and then implanted by IVF. The mother is infertile and unable to carry a child. We get a donor egg inseminated by the father and then a surrogate, so a completely other woman will carry this egg. If both parents are infertile, but the mother is able to carry, the donor egg is inseminated by sperm donor via IVF, and the embryo is transferred into the mother. And then in vitro fertilization specifically, mother is fertile but unable to conceive, mother is infertile but able to carry a child, father is infertile and mother is fertile but unable to conceive. All kinds of situations why people would use IVF. IVF is the treatment of choice for older women. It's used to treat female infertility. The age of the eggs, not the reproductive system, is a factor in infertility. And age-related infertility is treated by using IVF and eggs donated by younger women. So a lot of younger women will sell their eggs. It's a big industry. These companies will pay a lot of women a lot of money for young women's healthy eggs, and older women will use them to fertilize in a dish the young woman's egg and the father's sperm, and then insert it into either a surrogate or the mother whose eggs are older. So performing IVF, gametes are retrieved, fertilization occurs in vitro or outside of the body. The embryo is grown in the lab, and then the uterus or the embryos get transferred to a woman's uterus, whether it's the mother because she can carry or a surrogate because the mother for some reason can't carry. Intracytoplasmic sperm injection is a variation of IVF. The sperm is injected into the egg, and it's for the treatment of male infertility. So you got to get a healthy sperm somewhere and then fertilize the healthy egg. Gamete intrafallopian transfer is also an option. The transfer of gametes to the oviduct and then fertilization occurs in vivo in the body. It doesn't occur in a petri dish. It actually occurs inside of the woman's body where it's supposed to. This is less common, but it does happen. Zygote intrafallopian transfer. Zygotes are created by IVF and then transferred to the ovid. Little circles right there. Surrogacy is another form of ART. Egg donor surrogacy, IUI of surrogate with intended father sperm. The surrogate is both the genetic and gestational mother. Or there's just a gestational surrogacy where the embryo from the intended parents is transferred to a third party, a woman, and the surrogate has no genetic relationship to the child. There are, of course, moral, ethical, and legal questions surrounding ART and some of the risks involved. There's a higher incidence of multiple births, which you will talk about in your discussion this week when you learn a little bit about Octomom if you've never heard of her before. There's a higher risk of ectopic pregnancies when the egg and the sperm fertilize too early and it gets stuck. It doesn't go all the way down to the uterus. It gets stuck in one of the fallopian tubes, and that actually can be really dangerous for women. There's a higher risk for male children inheriting genetic disorders. Low birth rate is a risk. Higher risk of epigenetic imprinting disorders, higher risk of birth defects, and very high medical costs. All of these technologies are extremely expensive. Like thousands, tens of thousands, $20,000, $50,000 to do one round of IVF. It's insane. The next section is about genetic testing and screening. Of course, anybody can go get screened. Voluntary genetic testing. The carrier testing identifies individuals who carry a genetic disease and the risk of passing the disease to their offspring. Pre-symptomatic testing identifies individuals who may develop a genetic disease, maybe because it runs in somebody's family. Prenatal testing identifies whether an embryo or fetus has inherited a genetic disease. This is after fertilization, of course, and there already is an embryo. In all states, it is required for newborns to get tested for a variety of genetic disorders. All states test for the standard 3 to 8 disorders, but that's older technology. Some states have moved on to newer technology where they can test for up to 50 disorders. Pre-implantation genetic diagnosis. So performed on pre-implantation of IVF embryos. So while it's still in the dish, they look at it under a microscope like this. That is an embryo that has been fertilized and has barely started to divide. And DNA from a single cell is tested for the presence of a genetic disease. That is the earliest they can test it. Polar body genetic analysis. This happens before fertilization. So used in cases of heterozygous female carriers for X-linked disorders. We've talked about X-linked disorders and carriers before. The X chromosome with the mutant allele can segregate into the cell destined to be the egg or into the much smaller polar body. The polar body can be removed by micromanipulation. Don't need the polar body anyways. But we test that polar body to see if it carries the normal allele or the mutant, the diseased allele. If the polar body contains the mutant allele, that means the egg got the healthy allele, and that's what we want, of course, or what potential parents would want. Again, another ethical issue. The whole concept of savior babies. There was a whole movie about it. It's called My Sister's Keeper, if you want to watch it. It's a good movie. But parents have a first child. The first child has some sort of blood-related genetic disorder. So PGD is used to have a second child and to specifically select the traits that second child needs to have to save the first child, whether it's a stem cell transplant, a blood cell transplant, a liver transplant. They basically have the second child to save the first. So umbilical cord blood from the second child is used as a source of stem cells to cure the first child. That's the most common application. Traditional methods pose risks to the mother and the fetus, of course. Infection, hemorrhage, fetal injury, spontaneous abortion. These are all risks to prenatal testing. And not all genetic testing is accurate. Sickle cell anemia is a single mutation. It is present in all cases. So testing is very efficient because they only have to test. They know exactly where to test for that one mutant allele. Cystic fibrosis is a different story. There are 1,900 different mutations that have been identified for testing. If you can't test for all of those, it is very impractical. So it depends on the disorder you're testing for. Enzyme replacement therapy uses recombinant DNA technology to produce human therapeutic proteins for genetic disorders involving missing lysosomal enzymes. We talked about this a long time ago in one of the chapters. We watched a video about a boy who had the lysosomal enzymes and helped his disease. The problem is it treats the symptoms of the disorder. It's not a cure, and it's very, very expensive. While ERT is successful in controlling the symptoms, the ultimate goal is to cure genetic diseases. The most direct way to do this is by transferring a correct copy, a healthy copy of a mutant gene, to the body. This is called gene therapy. This involves the delivery of a normal version of a mutant gene to cells in the body, and once in the cell, the expression of this gene will produce a normal functional protein. That is the goal of gene therapy, of stem cell therapy. Gene therapy requires copies of cloned genes to be transferred. They need a delivery system in which they use viruses, which is very interesting. Viral vector. They also need the ability to identify the target cells in which the normal gene will be expressed. The normal gene needs to be inserted into very specific cells, not every single one of them, most of the time. Somatic gene therapy is a gene transfer to somatic cells to cure genetic disorders. Remember, somatic cells are every single other cell besides sperm and eggs. Certain types of leukemia, cancer, and type 1 diabetes have been treated this way. Genes are transferred into somatic cells of the body. The procedure involved only a single target tissue, and only one person is treated, and only after obtaining informed consent and permission for the treatment. That is a very important part. There are variations of gene therapy that are not yet used because we're still debating the ethics of them. Germline gene therapy, or cells that produce eggs and sperm, are the targets for gene transfer. After germline therapy, the transferred gene would be present in all the cells of the individual produced from the genetically altered gamete, including his or her germ cells. As a result, members of future generations will be affected by this gene transfer without their consent. Do we have the right to genetically modify others without their consent? Future generations? Can we make this decision for members of the future generations? Those are questions that are not easily answered. Enhancement gene therapy is another one. If we discover genes that control a desirable trait, such as intelligence or athletic ability, should we use them to enhance someone's intellectual or athletic skills? The FDA allows the use of growth hormone produced by recombinant DNA technology to enhance the growth of children who have no genetic disorder or disease, but are likely to be shorter than average adults. So this has already begun, but it's a very slippery slope from my child is not going to be as tall as the average person to I want them to be a basketball player. Where do we draw the line? Genetic counselors help people understand the occurrence and risks of having a genetic disorder in their family by reviewing medical facts, diagnosis, treatment, how heredity contributes to the disorder and the risk of having affected children. This is why a science background is very, very important, especially if you're going to have children later down the line. It is very important to figure out what runs in your family and what are your risks of passing that on to future generations. Alternatives for dealing with the risk of recurrence and how to adjust an individual with the disorder or the risk of recurrence. How to adjust to an individual, not the individual themselves. Who seeks genetic counseling? Pregnant women over the age of 35, because genetic disorders, and Down syndrome is a big one. The incidence rises very sharply in women over 35. Couples with a child affected by a genetic disorder or other disability, because it is likely their second child. If they keep having children, those future children are also going to be affected by that disease or disability. Couples belonging to ethnic groups with higher risks for genetic disorders. This is a thing. It's not racism, it's science. There are diseases that run through different ethnicities differently. Consanguineous couples, those are couples that are related to each other. Individuals whose environment may pose a risk to pregnancy. Women who have had recurrent miscarriages or infant deaths will seek genetic counseling. Why do I keep having miscarriages? Why can I not carry a baby to term? Couples who believe they may have or carry an inherited disorder, something that runs in families, should always know this. Pregnant women are at high risk for birth defects, as diagnosed by other methods. Individuals with a familial history of genetic disorders often seek genetic counseling. That is all for chapter 16. See you all next week.